Yeah, so similar to Vera, I also looked up Rigor and standardization in Google, and I did see ISEV predominantly there. The reason probably is most of the people just dwell around rigor and reproducibility, which is what they try to do by doing rigor and standardization in ISEV anyway. So again, for the topic again here, so we've been hearing about this concept of unable to reproduce scientific data for quite some time now. That's been a major talking point, especially from the funding bodies, and we as researchers have the onus onto us to try to do something about it. And that's coming through ISEV, and what we try to do here is, in a way, try to make these reporting as well as transparent, as well as making these experiments as rigorous as possible here. So there's 2 points towards this when you're trying to achieve the concept of reproducibility in a way. So one, I would say, academic integrity that comes within the researcher. The onus is completely on the researchers. And the second part, in the context of omics, would be on transparent reporting. So what I mean by transparent reporting and what I mean by why you need this, I will come to that on later parts. So again, I'm sure this has been touched upon in the morning session by several speakers, because what you isolate pretty much is what you work with. And again, the EV isolation or the EV enrichment technique that you use is going to decide what cargo in terms of protein, RNA, metabolites, or lipidomics you're going to identify, right? So if you end up isolating EVs in a crude way, obviously you're going to identify a lot of non-EV material in your sample, and that is given. And I'm not going to dwell into the isolation methods here, but I would just like to emphasize on this point that based on the question that you're asking, you may need to choose a method of choice that is probably going to enrich for your EVs in a better way. So before dwelling into that, when you come to comparison of EVs and when you're trying to use any of the omics techniques, one would normally try to, again, these are comparison between EVs, and very rarely we do see studies which is profiling one EV sample at a time, but rather we would try to do a comparative analysis in most cases. So in that case is what you do. So you normally normalize by total protein. That's what you normally see. Most of the researchers normalize their EVs based on total protein amount, but also you can normalize by total number of EVs, right? So again, the protein concentration, the RNA concentration can change. You can also normalize by volume or the number of cells. So the most preferred way one would do is normalize by proteins. So this is one example that again is an unpublished data. So you can see here, the gist here is we're looking at the role of this E3 ubiquitin ligase called NET4 in the biogenesis of EVs. And we knocked out NET4 using CRISPR-Cas9 in a colorectal cancer cell line, and we can see that impedes the secretion of EVs, right? Saying that ubiquitination is important for the biogenesis of EVs. But when we do a Western blot by equal cell number, we are seeing that the ALIX loading into the EVs is dependent on this E3 ligase NET4, right? But if you do this, like for example, if you're doing a mass spec analysis by loading 30 micrograms or 20 micrograms or equal protein amounts, you will miss this observation where you may end up seeing that ALIX is indeed secreted in similar levels in some cases, but in some cases you still see that in mass spec depending upon the question that you're asking or the protein of choice in terms of the biogenesis. So in a way you may need to normalize by different methods. The questions that you're asking, you may get better answers if you're normalizing by the total protein amount, equal cell number, equal volume, as well as equal number of EVs. And also one needs to be mindful about the EVs in terms of the surface area that we are working with. Now, even though we are trying to normalize by protein amount or number of EVs, you need to be mindful that if you're putting equal number of EVs, you're working with, let's say, large EVs versus small EVs, you have a higher surface area for large EVs. You tend to have more protein with the assumption that there's more protein packed or equal amount of protein packed into these large and small EVs in a way. But when you take into the small EVs here, you would tend to have more membrane content, the lipid bilayer content in them for the total amount of protein that you're normalizing. So you would normally identify more membrane proteins in that structure, right? So you would need to probably have a think about this prior to doing any analysis, especially in the context of bulk studies such as omics. So I'm going to predominantly focus this talk on proteomics because given my expertise lies on protein chemistry, also with 15 minutes I'm not be able to cover all the other topics as well. So again, this also need to be taken into account when you're looking at for the functional assay, as well as you may need to think about how many EVs does one cell secrete, and that is the context of small EVs versus large EVs, and when you're trying to compare between the functional consequences of these EVs onto the recipient cell, right? So again, reproducibility of EV isolation. This is a question that we all ponder upon, again, between people in the lab, and then when you talk about interlabs, there's always a lot of issues within the reproducibility of the data, and that can be compounded when you're probably doing a high throughput analysis like proteomics with this data, because I'm sure most of you would have wondered, right? You are also working on the same cell line, isolating the EVs, and you go to the literature and probably download the data from another group. They have also isolated EVs from the same cell line, but the content is not always the same, right? And again, this all comes in terms of there's various factors which can decide that. And when you try to do that within the lab, you need to be careful in terms of trying to reproduce that. So how much of you have thought about this, like in terms of, I'm assuming that many of you would have grown cells in lab. Do confluency of cells affect the EV cargo, right? So now we do know, based on my training in cell biology and biochemistry, that confluency can change some of the membrane protein expression. Would that change EV protein expression as well, right? And what about the other factors like CO2, growth factors, the FCS, starvation, stress, and mycoplasma, right? So we do know the answer for this question is all of these indeed affect the cargo at a certain level. So all these growth factors, all the external factors can change the cargo. And were you able to validate the content, like proteins or RNA that you characterized by a high throughput method, by orthogonal methods? Meaning if you've identified an RNA, have you tried to validate that by qPCR or a proteomics then followed by a Western blotting, and it struggled to identify that, right? And there are several factors for that as well. But again, the reproducibility of your EV isolation is important for all these aspects, right? So in terms of proteomics analysis of EVs, there's three factors here. So you may need to think about, like similar to what Veera said about the calibration and the sensitivity of the instrument. That again applies for the mass spec as well, where one needs to be mindful about how it is calibrated and whether it is sensitive enough. And that predominantly relies on the co-facility or your collaborator or even the proteomics guys in the audience who are trying to run the sample. The second part is about the sample preparation. Again, the reproducibility is a key here. You're trying to establish methods in terms of trying to prepare the sample for loading into the mass spec. I'm not talking about EV isolation here. It's rather the steps that follow after you isolate the EVs in terms of preparing your samples for mass spec. And the last part again is the data analysis, and here again one needs to be careful, because if you change few things here and if you're not reporting these things transparently, again one may not be able to reproduce the same results that you got even when you're doing a bioinformatic analysis later on. So again, when you talk about mass spec-based proteomics, you can do analyze your samples by various methods. You can use a label-based method where you can use many of the metabolic labeling methods like SILAC, where you grow the cells in light, heavy, or medium isotopes, where thereby you're labeling the proteins which are going into the EVs with different isotopes. Thereby you can quantify them by mass spectrometry. Or you can do this by chemical labeling or even the enzymatic labeling. All these things happens after you culture the EVs, isolate the EVs, and then you can do the post-processing steps here, right? Likewise, you can also do the label-free method, which is also after you isolate the EVs from your cells or your plasma or your tissues, and then you can subject to your mass spec analysis. So again, we are not here to tell you like what is the way that you have to do, because again, the method of choice of labeling, the method that you're using for quantification is completely up to you. But the one thing that you can do perhaps is transparent reporting, right? So that's the key here. It's not about if you want to try to reproduce the results. So now this is what you would normally expect, right? This is what normally we all aspire to do, because again, we do produce a heat map, we produce a volcano plot, and then see, okay, these are the red ones are the most significant ones which are significantly dysregulated between your conditions, and then you try to get a Pearson correlation which is more than 0.9, right? So that would normally, you would say the runs are pretty close. And at this age where the sensitivity of the instrument is quite high, we would expect that most of the runs you would get more than 0.9. This is what I mean by the runs is you're doing biological replicates, you're loading them onto your mass spec, and then you're getting the results, and then you're plotting the LFQ intensities of the proteins side by side, and you're seeing whether you're able to have a good or a high correlation between your runs. This becomes a problem, or I would not say a problem, rather a challenge in terms of when you're trying to working with understudied or non-model organisms. Like when you work with human mice, rat, it becomes a bit more easier. Believe me that even now the protein or the genome annotations are going on, and still people are refining the proteins that have been identified in the genome or characterized in the genome. Still people are identifying new open reading frames in them which have not been characterized before. But this is even huge for non-model organisms. In that case, what you need to do is probably rely on genomics or transcriptomics data, try to convert that into your protein database and try to do a search, as well as use model organisms and try to do your MS data search onto them and then try to do the analysis afterwards. But however, the caution here is when you're using large databases, when you try to maximize your results, when you're trying to identify more proteins, then you may create more FDR, false discovery rates, and you need to be cautioned about it. Here you can see IDs from Decoy. So Decoy are again non-standard databases, and you may get more identification here. So if you want to get more protein identifications from many of these samples like the plasma and others, you may need to, as I said, you can use multiple enzymes here like trypsin and others. You can do better fractionation, longer gradients in the mass spec, and you can also do more fragmentation techniques. And also other issues like keratin contamination, too much enzymes in your sample prep, the correlation between your technical and biological replicates needs to be reported. So with that, I'll probably finish my talk with this Proteomic Standards Initiative, which is called the MIAPI, which is the minimum information about a proteomics experiment, where you need to report what you have done in terms of your proteomic experiment. And this is what is mandated by the proteomics community, right? But however, this I'm sure most of you can relate to, right? So this is from this paper, the Proteomic Standards Initiative. After 20 years, what they've done, they're saying it seems that most researchers would like others to provide rich metadata, but they are reluctant when asked to provide all the information about their own data sets and avoid systems which required to do so, right? So which again, I'm sure we can all relate to, but however, I would encourage everyone to report as much as possible in their manuscripts so that can be transparent. With that, I'll stop, and thanks to the audience.